the body will not accept them. When I say the body will not accept them, I'm saying that there are receptors that are on the surface of cells that viruses have to attach to in order to get inside of the cell. These receptors, and we'll talk about that a little more when we get into the melanin piece, but these receptors have to recognize the virus. And it looks at the protein coat of the virus in order to identify if it can come into the cell or not. Okay? Bovine viscivirus won't do that because it is a cattle and sheep virus, and that does what? Cannot cross the species barrier. So what you need to do is take this bovine viscivirus out of its shell. So you cut it, you use some recombinant DNA reagents, and you take this bovine leukemia virus DNA and RNA out. You take it out of the shell, and then you give the body something it can recognize, like a herpes virus. So the herpes virus will take inside of a human being. So herpes is a virus that is similar to chickenpox. So the virus protein coat, once it gets to the receptor sites in the cells of the human being, it will recognize that as a human virus. But we don't want to give the patient herpes. We want to give them bovine viscivirus or A.I.D.S. So how are we going to do this? Well, what we do is we cut using recombinant DNA reagents. We cut open that herpes virus shell, take out the herpes virus. We take out the herpes, we discard that, and then we inject into that coat the bovine viscivirus. Okay? I hope you can follow that. So what we did was, essentially, we created what is called a Trojan horse. And you know, in Rome and Europe, you heard of the Trojan horse philosophy. So we've taken a bovine viscivirus, cut it open, extracted out the DNA, right? We've taken that DNA, cut open a herpes virus, extracted out the herpes DNA, and put into that herpes coat the bovine viscivirus. So now we have a herpes coat that in the human being will be accepted, but inside of that herpes is bovine viscivirus, which will cause HIV/AIDS once you put it into a human being. So we inject this newly formed recombinant bovine viscivirus wrapped in a herpes coat into a human being. And now, when this is injected into a human being through the vaccination process, the HIV/AIDS, or bovine viscivirus will take, causing symptoms of what? Remember we said bovine leukemia virus is a T-cell attacker, right? T-cell attacker that will present symptoms of immune depression. That's how you get bovine leukemia virus to take in a human being. All right? Now, the human being will get sick. The symptoms will be both symptoms of sheep viscivirus and symptoms of bovine leukemia virus. All right? Now, we talked about the receptors, and I want to go into this melanin piece really quick with y'all, real briefly. And a lot of this, I'm kind of going fast. I can give you a more detailed presentation, but I just want the common folks to kind of get an understanding of how this thing works. And we can go into, contact us on the website, and we'll give you more information on exactly how this works. When we make this in the lab, these are the exact steps we go through. I'm just throwing out all the scientific nomenclature, because a lot of times when you throw that in, it gets people confused. So I'm keeping this as simple as possible for the common person to get it. Now, we talked about human cells. This is a cell, has a nucleus in it, ribosomes, and all that stuff. This is a human cell, okay? Okay. On the surface of human cells are these things called receptor sites. And they're these long little things, and they go just like this all around the cell. I hope you can see that. These receptor sites have very unique properties. They determine what gets into a cell and what gets out of a cell. So receptor sites are like lock and key mechanisms. In science, we call them docking proteins. The receptor site may be built like this, and then the protein that docks to it has to fit in. So it's a glove fit mechanism that the protein or the virus has to have a specific shape to lock on to that receptor. And that's what these receptors are, okay? So in order to get into a cell, you have to get into the docking protein or the docking receptor site in order to get into the cell. Anything, whether it's nutrients, whether it's a signal, all the things that the body uses to send a signal into the cell has to go through the docking proteins or the docking receptor sites, as we call them. So the receptor sites play a role in getting in and out of the cell. So a virus, if we have a virus here, like we just put in, which is now the bovine, right? Visna virus. We got this bovine Visna virus floating around here, and on the bovine Visna virus, it's trying to attach on to this receptor. And now, remember, the receptor wouldn't have recognized this if it was just bovine Visna virus shell. It recognizes it because now it's a herpes shell around here. And that's how it's able to get into the cell. Now the virus isn't this big. I'm just drawing it big enough for y'all to see it. The virus is much smaller than a human cell. But I just want you to see that. So the docking protein will lock on to the herpes coat, which cloaks the Trojan bovine Visna virus in it, and that's how to get into the cell. Now what does this have to do with HIV/AIDS and melanin and black people? Well, just so happens that the receptor sites that are in people of color, the melanin facilitates the types of receptors that are on the cell in the human being. So in black folks or African people, we have a certain type of receptor. And this receptor on the allele, when we look at the chromosomes, is called delta 32 positive, or CCR5 delta 32 positive. That's what this receptor is called. Let me break it down a little further for you. CCR5 delta 32 positive. This is an allele that codes for this receptor site, okay? These receptor sites for CCR5 delta 32 are more abundant in people of color than any other race. So much so that people who are Caucasian who do not have this CCR5 delta 32 allele in their DNA are more resistant to catching HIV infection. In other words, if a white person who has CCR5 delta 32 negative, which is a mutation of this one, remember we have mutations that occur in our DNA, delta 32 negative, let me put it under here, a delta 32 negative allele, if they have two homozygous traits for that, they will not be able to catch HIV even if you give it to them. But in people of color, this CCR delta 32 receptor sites are more abundant. Well, what does this mean? Well, what this means to you all is HIV/AIDS really has a strong affinity to this CCR5 delta 32 allele, which brings into question, is the virus racist? And viruses cannot be racist unless they're designed to be that. So how does AIDS know that this is the virus it's supposed to attach, this is the receptor that it is supposed to attach to in order to get into the cell? How does HIV know that? Well, it either was designed to know that or manipulated somewhere in the lab to know that it must bind to this receptor site. If it does not bind to that receptor site, it's much more difficult to get into the cell. In some cases, cannot get into the cell unless it binds to this delta 32 positive allele.